And yeah, that's pretty much it for this page. If we speak now about the immunology chapter, let's go back to it. Basically, as I told you, from biochem, every class I'm going to give you a biopsy from it. So every class I'm going to explain one page. But by the time we finish immunology and endocrinology, we'll be halfway through, and I will jump directly to finish the rest of it. So it will be an easy sale when we do that. So when my plan is like until the end of this month, December, we'll finish biochem, finish immunology, finish endocrinology, finish GIT. So we have much less material left. We will be done with around more than 40%, which is my plan, okay? So let's go back to our friend, immunology. So an antibody. An antibody is responsible for defending you. Is it against bacterial or viral infections, guys? Well, mainly at least. An antibody is responsible for protecting us. Is it against bacteria or viral? Mainly. Bacteria, yeah, that's correct. So the antibody structure and function. To start with the structure, we have something like this. Well, you have something called the Fab fragment. And what is a Fab fragment? Look at this antibody. It has this region and it has this region. Well, the upper region here, we call it a Fab region, okay? While the lower region right here, we call it what? We call it the FC region. So Fab region, FC region, Fab region, FC region. This is super important. So, yeah, so the Fab region have light chain and heavy chain. You are like, light what? Light chain and heavy chain. See this one that is not attaching to anything? Yes, this is a light chain. And can you see this one that is really long? This is a heavy chain. So we have a light chain and we have a heavy chain. Yeah? So far we have light chains, we have heavy chains that work to recognize the antigen. Well, who recognizes the antigen? Is it the FC portion or the Fab portion? The answer is the Fab region. Do the antigen bindings, has the antigen binding site. So if you get a question, where do you identify the antigen? The answer is at the Fab region, okay? So while the FC region, which is the back region right here, FC region of IgM and IgG fixes the complement. You must know, and they do give you this question. This question will come to you like a drawing for you antibody like this, like the heavy chain and the light chain. And they will be like, oh, where does the complement bind? Is it 1, 2, 3, or 4? You must be comfortable that it binds to here, okay? Which is 2, yeah? So the complement binds to 2. You're like, how can I remember this? Well, guys, do you know what cervical mean? What does cervical mean? Can somebody tell me what does cervical mean? Cervical. Well, for me, I just remember like cervical means the neck. So the complement binds to the C, which is the cervix of the antibody, basically, to the neck of the antibody, okay? So yeah, it binds just right here. While the macrophages, I just imagine it as a giant M binding to both. They love playing with you in this one. They will give you, oh, is it binding to 1? Is it binding to 2? Is it binding to 3? Is it binding to 4? The macrophage. And you know that it is like this one, so you'll be like, it's 1. And your answer is wrong. You should say it's 1 and 2. Okay? It's 1 plus 2. They love to do this to you guys. So again, the Fab region binds to the antigen. This is the bacteria. This is the antigen that has the epitope on it, which is like the thing that helps you identify the bacteria. And then you have this complement binding site, which is located at the FC region, and you have the macrophage site, which is located also at the FC region. Okay, so yeah, light chain contribute. So what did they write here? Heavy chain contributes to the FC and the Fab region. Let's take a look. This is the heavy chain, yeah? So, and this was, what? What was this region? That was, what do we call this region, guys? The whole thing, from here to here. What do we call that? This is the Fab region. Yes, this is the Fab region. So as you can see, the heavy chain take a part and make you a part of the Fab. Yes. While and also the heavy chain, which is basically this thing, this whole thing, makes you also all of the, well, how do we call this portion, guys? How do we call this portion of the antibody? The FC region, exactly. So we call it the FC region. So the next thing, here we have done the Fab region, we have done the FC region, and now we can move forward. So light chain contributes only to the Fab region. So the Fab region was only here. The light chain is only within it. So the light chain only makes you the what? The Fab region. That's it. Okay. So now you are, guys, kind of comfortable, kind of confused, but the main idea now for you, if you understand the words I'm going to tell you right now, that means you understand everything that is needed from you, which is light chain, heavy chain, Fab region, FC region, complement binding here, macrophages bind to both because it has two legs, yeah? It's an M and it has two legs, so it binds to both. Okay, now let's add one more thing. So we have something called basically a constant region, which has little c's here. C, 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 C. Well, why this is important, we'll see in the future. But why, if you have a constant region, that means that you must have what? A variable region. So where is the variable region? Here it is. So does it mean that the variable region can change its structure? Absolutely, it can. It can change its structure. And we'll speak about what and how it changes its structure and what the change in structure means. So we have here the following. Fab is a fragment antigen binding. So yeah, that's where the antigen bind. Where does the antigen bind? You must know that it's to the Fab, yeah? And it determines the idiotype, okay? What does idiotype mean? Well, you will only have one antigen able to bind that antibody at that specific location, which means, what I'm telling you, that the B cell, this is a B cell. Does a B cell make antibody or has to convert to something else? Should the B cell secrete antibody directly, or it should make something else, and that something, it has to convert. Convert to what exactly? What does it convert to? To what secretes? Yeah, exactly, a plasma cell. Excellent. So a plasma cell. So that plasma cells make the antibody. I want you to know, for each B cell, it makes a distinct antibody from the others. There is no two B cells that make the same antibody, okay? So that B cells make antibody against COVID. That B cells make antibody against influenza. That one makes antibodies against, I don't know, well, Streptococcus, pneumonia. So each one is significant and specific for one antigen, one epitope. That's it. So for each one of them, they have only one form of receptor. So only one antigenic specificity expressed per B cell. Okay? So each antibody is able to bind to only one antigen. Okay? If we speak about the FC portion, FC region here, where we have the complement binding and where we have the macrophage binding, notice that it's constant. What does it mean, constant? It means that for different types of antibodies from the same group, which I mean, like, for example, You have IgGs for influenza, COVID, strep pneumonia, and Staph aureus. All of these antibodies will have the same Fc region. Yes? So if all of them have the same Fc region, just take a wild guess. What will help you more in it? Would it be if they just bind to something totally new and they bind to it, something totally new in the body? We had something, actually, let me just tell you, we had something called the natural killer cells, and it can bind to it. And it binds to it using something called the CD16. And that will lead to secretion of the gran enzyme, which will destroy this bacteria or this antigen, basically. Okay? So let's speak about here. Other than the fact that it's constant and it does not change from one antibody to antibody, it also does have carboxyl terminal as like basically a chemical structure ending. Also, it participates to the complement binding. What do I mean complement binding? If you take a look here, the complement binds here. So you're like, what is a complement? Well, a complement is a humoral system that I will speak about today in details, and it helps basically defend you against a lot of types of bacteria, okay? By creating some ways to protect you, either by calling other immune cells or by directly destroying the bacteria. Okay, so also these have some carbohydrate side chain, and they also have, oh yeah, by the way, you need to know about this disulfide bound. See this disulfide bound? It has this, it forms at the hinge. Please know about the hinge, because in the near future I will speak about some pathology related to it. So, and yeah, this area here, it tells you if the antibody you have, is it an IgM? Is it an IgD? Is it an IgG? Is it an IgA? Why? Notice here on this side, an IgG looks like this. An IgA, see, at the Fc portion, it was connected by the J chain, which make it look completely different. So it determined, is it an IgG or an IgA, the Fc portion. The Fc portion also can make you an IgM. Guys, between these three, which one is the smallest? Is it IgG, IgA, or IgM? Which one of these antibodies is the smallest? It's the IgG. So if it is the IgG, think about it. If it's that small, that means it can pass it through the placenta. So if they ask you which antibody passes through the placenta, it's IgG, okay? Because it's the smallest. Okay, let's go back here. So here we have generation of antibody diversity. How can we make antibody against COVID and make antibody against influenza and make antibody against, I don't know, well, Streptococcus pneumonia from the same antibody? Well, basically, you need to change which region? The variable region. So you play with the Fab region, basically, the variable region added. How do we do that? Well, you have to do something called Vdg recombination. So Vdg recombination. Sadly, in first aid here, it's mildly explained. You don't have to understand it, actually. You just need to know that it exists. I will explain it, but if you don't understand it, it's not required from you guys, okay? So, basically, here it is. Yes. Okay, so I want you to notice the following. Much better. If we speak about this picture, you can see that here you have this basically locus, which is located in your genome, okay? It has VDG. So we have something called the VDG recombination, yeah? So according to this VDG recombination, you can make this area of your immunoglobulin highly specific to one antigen and not the other antigen. How they do that? Well, they are like, okay, what does influenza have? They are like, well, it has this A antigen, for example, and it has this B antigen, and it has this C antigen. Well, what do you guys have? Well, we do have A, Z, W, F, whatever, weird numbers, you know? So you need only to get the A, B, and C. So how do you do that? You do remove these ones by splicing, okay? You splice them out by a process I will tell you in biochem. We splice out gene by a process called spliceosome, okay? And that kicks out some genes. So you will have some genes left. So these genes that are left, well, look what happened. Now you have, what just happened? You had the D and the G has just connected. Excellent. So the D and the G has now connected. So what should I do now? You should add the V to them. Should you add the whole thing or only the portion you need for that exact virus or that exact bacteria? You add only what you need. Anything else, you throw in the garbage. So you will end up having only one exons that will be expressed. Remember, your gene is comprised of introns and exons. You only express the exons. Who decide what is an exon or an intron? I will tell you in the future. It's basically a sequence. So if you take a look at this one, this is the Vdg recombination. The main idea, if you understood nothing here, is to know there is a process called Vdg recombination that occurs at the Fab region of the antibody, and it determines the diversity of the antibody, okay? So if you look here, random recombination of the VJ at the light chain and VDG at the heavy chain. So all of this process is occurred at heavy chain and light chain, where at the Fab region. Okay, so we have decided how to make it like highly specific and make this antibody highly specific for that epitope. What should we do next? Well, you should make sure that you add some sequences to make sure that it will have high affinity to that region and it will not just, bind to anything else, okay? So how to do that? We have some nucleotide. Guys, what is a nucleotide? Can you remind me, please? We studied it today. At light chain, oh no, look, at the Vdg recombination, if you take a look at this picture, the first, there is this one that they are describing here. This process is occurring at the heavy chain, okay? But at the light chain, there is something only like the V with the J. But to be like truthful with you, you don't even have to know this process. So you don't have to go too deep in it. You just need to go deep enough to know that this Vdg recombination leads to the diversity of the antibody, and you need to know at which locus it's located, which is which region? The Fab region. That's what is required from you to know. That's what they ask in your word. Yes. So the nucleotide is it's a sequence of sugar, nitrogenous group, and a phosphate. Okay, so during the recombination, we start doing what? We add the nucleotide and we have this DTT. Why this DTT is important and why they even mention it here? It's just a random fact throwing at you, Why the hell I do need to know that? Guys, does anybody here have ever done hematology? He will know why the DTT is super important. Why the DTT is super important, guys? People who do hematology in their life. Come on. Okay, a bit disappointed, but it's basically no. It's the DTT is a marker. It's a marker of cancer, especially which type of cancer. the lymphocytic cancer. Why? This process is antibody mediated. So if it's antibody mediated, it will be at which cell? The B cell. So if it's the B cell, patient who have cancer of the B cells, what is cancer? It's hyperproliferation. So what is going to happen to the number of the TDT in case of hyperproliferation? It's going to be crazy high, yes? So we use the TDT as a marker to determine if this cancer is lymphocytic in origin or is it from somewhere else. So basically what I'm trying to tell you, that if the TDT will be high, this patient basically have a cancer with a B cell origin, yeah? So that makes it kind of easy actually, that you have just a marker, and according to that marker you can follow it and know that this patient has this form of cancer, and that's it. So if you see here we have lymphoid neoplasm, a lot of speaking, but I want you to notice the TDT, yes? So this DTT, here it comes again. Whenever you find it positive, it means, and that's why actually they mention it in immunology. In immunology, it's just a random fact. They are adding it there. But for the sake of hematology, you must know it. The TDT, when it's positive, it's a marker for lymphocytic leukemia. In the future, we'll be able to determine how to look at a vignette and know which type of cancer this patient has, okay? So let's speak now, after the DTT, what do we have? Well, after the DTT, we have random combination of heavy chains with light chains. Well, yeah, we have here the light chains and we have the heavy chains. Now we need to attach them to each other. Okay, how to do that? With disulfide bound, okay? This is not that important. Next, you need to make an antibody that is highly specific to that antigen, okay? So you need to make an antibody that first of all is diverse against a lot of antigens. That was the first 3 steps we spoke about. Now you need to make it against an exact pathogen. To make it an exact pathogen, you go to this variable region we spoke about and you do something called somatic hypermutation. Guys, I'm doing hypermutation, which basically silence most of the region except one small area that will be able to respond to the antigen. By silencing, which process of the following do you think I'm using? Is it acetylation? Is it methylation? Is it, no, enough. Is it acetylation or methylation? Yes, methylation. So I do the process to methylation to suppress the whole region except one small area that is specific for that exact antigen, okay? And then I do isotype switching, which occurs at the constant region, and that isotype switching basically make me jump between different antibodies and antibodies. Basically make me choose which antibody I want. Okay, do you want IgG? Okay, let's class switch to that. Maybe you want an IgD. Well, let's do it. So let's speak about how an antibody actually works. There is various ways an antibody works. One of the ways is in neutralization, which means an antibody just can bind to this pathogen and just paralyze it. If it binds all of its antibodies, can this bacteria bind to our cells? No, it cannot. They are just paralyzed, yeah? So they will be stuck. So if that is the case, they are stuck. They cannot do anything. So they will not bind. Done. You neutralize it. Okay, what about opsonization? Opsonization, I like to imagine it as a form of it's like if you have a dog and you bring a toy for the dog and tell him, hey, come and get it. Come and get it. You are opsonizing the dog, okay? So what do I mean by that? Well, you have the bacteria, yeah, inside of your body. An antibody comes and attached to it. The dog is basically the macrophage. The macrophage, when he sees that antibody, he's like, oh, my food. So he goes and engulf it and eats it. So that's antibody promotes phagocytosis. The last way is complement activation. How can an antibody, well, lead to that? Well, we know. Remember, guys, a complement can bind here and lead to some process that we'll speak about in length. But you also need to know that antibody activates complement as well. So it's activated by complement, but it can activate complement. Which complement exactly? We'll speak about BM making classic cars in a second. But for now, I just want you to know that antibody activates the complement. And when the complement is working, it makes a hole. How this hole is made? Basically, if this is the bacteria, complement start forming a structure. So one, subunit, 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 subunit. So it made a hole inside of the bacteria. So it start releasing the substances to the outside, which kill the cell. I just want to show you a really quick video about the MAC complex. Oh yeah, that one. Okay, take a look at this complement and how it works. So you have, yep, notice what has just happened here. Here is the complement. Different portions binding to each other. We'll speak about all of them specifically and which one they are. This is basically a pathogen. We want to kill this guy. So how to kill it? See, a ring has came and it has formed a bore-like structure here. Yes, it's forming a gate. And here we go. Start working. Done. So what it has done? Look. Boom. Explosion. Why does this explosion happen? There is two ways. Well, actually, there is three ways to do that. And the way that they lead to the explosion is actually creating a pore. When that pore happens, all of the substances from the inside start to go to the outside, which is like osmotic damage. And it does not happen gradually, slowly. No, just like boom, and it just explode. Okay? And when it explodes, you are done with the pathogen, which is a good thing. Okay, guys, we explained a couple of pages. Let's take 10 minutes break and come back after the break.